There are cells in the human body, trillions, 37.2 trillion cells in the human body, which is a lot considering it started off as one. Cells are the structural building blocks of all plants and all animals. Okay, they're produced by the division of a pre-existing cell. Okay, so you have one cell in the egg, the sperm comes, fertilizes it, that one cell divides into two, two into four, four into eight. So all cells come from pre-existing cells when the organism develops. Cells form all the structures in the human body. Everything's formed from cells. Remember our little chart going from least complex to most complex, molecular, and then starts with the cell. That's the lowest or the smallest form of life is the cell. Cells perform all vital functions of our body. Oh, why aren't we working again? Uh-oh. Why is my clicker not working? Oop, no, it went too far. Sometimes you get stuck on there. Okay, there we are. So all living organisms are composed of cells. All cells come from pre-existing cells. The smallest functional unit that displays the property of life is the cell. Cells are the building blocks of all multicellular organisms. That's what we're built from. There's two types of cells. There's sex cells, sperm in males, oocytes in females, and then everything else. Okay, so you have sex cells and somatic cells. Somatic cells are all the cells in the human body except for sperm and oocytes. Those are sex cells. So you have two cells, sex cells and somatic cells. A cell consists of cytoplasm. Cytoplasm is the inner part of the cell, which consists of cytosol, which is the liquid, and then organelles, which inhabit the cell or occupy inside the cell, and the plasmalemma. The plasmalemma is the surrounding cell membrane. Okay, so here's our cells broken down. It's divided into the plasmalemma, which is your cell membrane, and inside the cell is your cytoplasm. Cytoplasm is broken down into cytosol, which is your liquid, and your organelles, which are all the various organelles inside the cell. Okay, so organelles can be non-membranous or membranous. Membranous ones have a membrane. Non-membranous ones, organelles, do not have a membrane around them. So non-membranous organelles of the cell are your cytoskeleton, your microvilli, your centrioles, your cilia, your flagella, and your ribosomes. Those are all non-membranous organelles. And we'll go through each one of these two in detail. Okay, you can see what they're made of, the composition, and the different functions. This is your plasmalemma. Okay, this is a lipid bilayer. This forms your cell membrane. And it proves isolation and protection, sensitivity, support, control of entrance and exit materials from the cell. Okay, you have your cytoskeleton, which is your proteins and microfilaments. You have microtubules and microfilaments here. These make up your cytoskeleton. Okay, it gives you strength and support of the cell. Microvilli are extensions of the cell. Okay, they allow increased surface area for absorption and secretion. You got your centrosomes and centrioles, which allow the DNA to line up during cellular division to be able to split it into two daughter cells with the correct DNA going into both cells. You got your cilia, which are basically long microvilli. Okay, to increase the extension, and it causes movement of material over the cell surface. Let's take a alveoli, a lung cell. Lung cells have cilia. The cilia of a lung cell only beat in one direction. Okay, which direction they beat in? Perfect. They form things to go out of the lungs. They all move in the direction to force contaminants, pathogens, viruses, anything, and beat it out of the lungs. That's where you produce mucus and you cough it up. Okay? Those are your cilia. Ribosomes. Ribosomes take RNA and amino acids and make proteins. Okay? They're involved in protein synthesis. They're two subunits that come together and use RNA as a template to make your proteins. Okay? There's two types of ribosomes in the cell. There's fixed ribosomes, which are attached to your endoplasmic reticulum, and there's free ribosomes, which just exist on their own in the cytoplasm. Sometimes this doesn't go quick enough here. Come on. There it goes, and it jumps too far. Okay, we're back. So these are the front. You will probably see this picture again. That's a hint. Okay? Be able to identify these structures. And we're going to go through what all these structures are. This is a Golgi apparatus. It's like pancake system right here. These are mitochondria, where ATP is produced. These are peroxisomes, which digest bacteria, other pathogens. These are the nuclear pores. These are holes in the nuclear membrane, which allows DNA, go to the RNA, to transfer out of the nucleus into the cytoplasm where it can make proteins. So you have nuclear pores in your nuclear membrane. This is smooth endoplasmic reticulum, which is endoplasmic reticulum with no ribosomes. Ribosomes are these little dots. So when it does not have any dots, this is what's called smooth endoplasmic reticulum. Rough endoplasmic reticulum has dots. These little dots are ribosomes. I'm blanking on words. The little dots are the ribosomes. So when ribosomes are attached to endoplasmic reticulum, it's called rough endoplasmic reticulum. When ribosomes are not attached and there's free floating, they're called free ribosomes. When endoplasmic reticulum has no ribosomes attached, it's called smooth endoplasmic reticulum. So there's your smooth endoplasmic reticulum. There's your rough endoplasmic reticulum. These are your fixed ribosomes attached to endoplasmic reticulum. And these are your free ribosomes. These little dots are there floating around in the cytosol. All right, plasmalemma is your plasma membrane. It's a double lipid bilayer. Your cytoskeleton are your microtubules and microfilaments that make up the structure, hold the cell as a shape. Your nuclear envelope, which surrounds the nucleus. Again, so the nuclear portion of the envelope. This is the nuclear envelope. Okay? You have a nucleolus, which is dead center of the nucleus. You have nucleoplasm. This is like cytoplasm of the cell. This is like cytoplasm of the nucleus. So it's called nucleoplasm. You have chromatin. This is your DNA. Now, DNA is normally all spread out. Okay, it's not confined into chromosomes yet. When the cell replicates, we condense it into chromosomes so we can split it apart and make two cells. So normally DNA is chromatin. It's spread out, it's all loosely combined. That way you can open it up and unzip it and allow you to transcribe it and translate it into messenger RNA. And we actually can use it. So it exists as chromatin. These are your centrioles and centrosomes used in cell division. Okay, these are your lysosomes. Lysosomes are just like peroxisomes. They help dissolve pathogens and other things in the cells that shouldn't be there. The only difference is peroxisomes specifically dissolve hydrogen peroxide, whereas lysosomes dissolve a whole bunch of other things. This is your cytosol, which is your liquid. Okay. These are secretory vessels. These break off from the Golgi apparatus. The Golgi apparatus processes your proteins, and then when the process is going through this whole system, it breaks off into these secretory vesicles. These secretory vessels go to the merge with the plasmalemma and secrete either the hormone, the protein, whatever, into the bloodstream, into the extracellular fluid to get through the rest of the body. And then these are your microvilli. Microvilli increase the surface area. Okay, that way they can absorb a lot more and secrete a lot more. It increases surface area, that's what they do. So microvilli are the small ones, the cilia are your larger ones. Okay? Any questions on the cell? We're going to go through each one of those individually too. But know this picture. Again, that's a hint. Computer is not responding very quickly all of a sudden. There we go. Okay, so membranous organelles of the cell. The membranous organelles are your mitochondria, your nucleus, your endoplasmic reticulum, your Golgi apparatus, your lysosomes and peroxisomes. All those structures have membranes around them. Okay, this is the composition and function, which we're going to go through here. We talked about a little bit. Again, lysosomes and peroxisomes are very similar. Again, both contain digestive enzymes, but peroxisomes are specifically designed to break down hydrogen peroxide, which is toxic to cells. Okay, let's go through each one here. So the plasmalemma. Plasmalemma is your cell membrane. It's composed of phospholipids, glycolipids, proteins, and cholesterol. That's what makes up your cell membrane. Okay. The cell membrane, it's also called a phospholipid bilayer. It's two layers of phospholipids that come together. Major functions is physical isolation. Isolates the cells from other parts, other things in the body. Regulation of exchange, allows certain things in, allows certain things out. Sensitivity, response to the environment stimuli. Allows cell-to-cell communication through adhesion to structural support. Cell-to-cell communication is extremely important. Take the heart, your muscle cells. Your muscle cells have to communicate with one another. Once one starts to contract, all have to contract. Every muscle cell in the heart has to contract at the same time for the right atrium, right ventricle, left ventricle. They have to contract together. They have to talk to each other. So cell communications is extremely important for a lot of reasons. Okay, it's a phospholipid bilayer. It's composed of 2 layers of phospholipids. Phospholipids have a hydrophobic head at the surface, inside lining and outside lining. Have a hydrophobic fatty acid tail facing towards each other. Okay, this is your phosphate group, and these are your this is your fatty acid tails. This is your fatty acid tails, and this is your phosphate group. This makes up your membrane. Fatty acid tails, phosphate group, fatty acid tails, phosphate group. That's how your membrane's composed. So this is a negative charge, and these don't have charge. These are ionic. So this is hydrophilic. This means water loving. So outside the cell, this is your extracellular fluid. This is your intracellular fluid inside the cell. Okay? So these parts are hydrophilic. They love water. Inside and outside. Inside the plasma membrane, these are hydrophobic. I can't write really well with this pad. So hydrophobic. This is water hating. So this is how your cell membrane is composed. It's a lipid. These are lipids with phosphate heads on them. So it's a lipid bilayer. There's 2 layers. The inside layer is your fatty acid chain. Okay, that's hydrophobic. It does not like water. The outer layer is your hydrophilic phosphate group. So that's your phospholipid bilayer. That makes up your plasma membrane, your cell membrane. There's a hydrophilic at the surface. Those are your inside lining and outside lining. Okay. So in the extracellular fluid and the intracellular fluid, you got your hydrophilic layer, your phosphate layer. The hydrophobic layer is your tails, your fatty acid tails. Those face each other on the inside like I drew. So the outer layer consists of glycolipids and glycoproteins. Glycolipids and glycoproteins form what's called a glycocalyx coating that coats the layer on the outer surface, okay, where your extracellular fluid is. Your inner layer does not contain glycolipids or glycoproteins. So the glycocalyx is located on the outer surface of the plasma lemma. Come on, slide. I don't know why it's not behaving right. It kind of skips sometimes. Okay, this is a better drawing than I drew, okay? You can see if you can follow my mouse. This is a hydrophilic heads. These are your phosphate groups. These are the hydrophobic tails. Hydrophilic means water loving, hydrophobic means water hating. This is cholesterol embedded within the fatty acid chains. Cholesterol is hydrophobic, okay? It's a fatty acid. Okay, so this is a lipid bilayer. On the outside of the cells, you can see the glycoproteins and the glycolipids for what's called the glycocalyx. These do not occur on the inside of the cell. So if you follow my mouse again, this is the extracellular fluid. Okay, this is the intracellular fluid. Okay, what separates the extracellular fluid from the intracellular fluid is the bilipid layer, the plasma lemma. And they're arranged with your hydrophobic, the fatty acid tails inside, and the phosphate heads on the outsides, both extracellular fluid and intracellular fluid. Okay? And the glycocalyx is only found on the outside extracellular fluid, not on the inside. Okay? Oh, come on, computer. I don't know why it's skipping like that, not skipping certain ones. So you have structure of the plasma lemma is composed of protein molecules. You have peripheral proteins, which attach to the glycerol portion of the fatty acids, and integral proteins, which is embedded within the cell membrane itself. These forms little channels like calcium gates, sodium gates, and allows glucose and things to be brought into the cell. Those little channels you saw in the previous slide, right in here, you can see these channels set up, and things can move in and out between the cells and not worry about the fatty acid chains. So ionic compounds can pass in and out through these channels. They're called gated channels. We use that a lot in nerve transmissions. I mean, how many people have been to the dentist and they'll get an injection and your lip gets numb, right? All that doesn't stop you from feeling pain. Doesn't get away with, doesn't dissolve the pain at all. All it does is block salt. That's all local anesthetic does. Sodium, potassium change place. That allows a nerve impulse to travel. Okay, if we block that sodium through those gates I just showed you, local anesthetic blocks those gates. Sodium and potassium can't move. The nerve it can't transmit the nerve. So the pain's still there. The nerve still feels it. It just can't transmit it. So all a local anesthetic does is block salt. It's simple. That's how it works. It's kind of cool. Okay, this is composed of sterile molecules like cholesterol that are inside the cell membrane. Okay, maintains fluidity of the membrane, allows it to flow and allows the cell membrane to move. Membrane permeability, there's different processes of bringing things in or out of the cell. Okay, a passive process, passive means does not require energy. Diffusion, osmosis, and facilitated diffusion. Active processes, active means it requires energy. Active transport, endocytosis, exocytosis. Those are 6 ways of getting things into or out of the cell. Okay, passive process diffusion. How many people ever made Kool-Aid? You take a glass of water or a bowl of water and you pour this packet in there, like purple, red, whatever color it is, right? And all the colors in one spot when you pour it in, right? And then over time, what happens? That color dissipates through the liquid. That's diffusion. Very simple. It's a high concentration of your solute, which is your local, and then it spreads out. That's diffusion. And it happens with no energy. It's going to do it naturally. The second law of thermodynamics, things pertaining towards entropy, towards randomness. And randomness is not one column, so it all spreads out. So it does it without energy. So it's a movement of molecules from an area of high concentration to an area of low concentration. Okay, just think of Kool-Aid. Permeability, concentration gradient, molecule size, charge, temperature, all affect how fast those move. Slower ones are going to move faster than other ones. Ones with a charge, if it's in water, are going to move faster because water is polar. Okay. Concentration gradient, the greater the gradient, the more concentrated that you pour in the water, the faster it's going to spread out. Temperature. What happens if you heat the Kool-Aid? It goes a lot faster, right? That spreads out a lot quicker. Okay. Small inorganic ions and small molecules are involved in diffusion. And you're not going to be click. It's probably going to go fast. Okay, so this is diffusion. This is your cell. Okay, diffusion is the movement of molecules from high concentration to low concentration. Okay, the difference between the concentration of the outside of the cell and the inside of the cell is what's called a concentration gradient. Okay, which way the little gray dots are CO2 molecules? Is the concentration greater inside the cell or outside the cell? Inside, right? So it's, where is it going to flow? The concentration gradient is going to go from inside to outside. That's where it's going to move. Okay? That's diffusion. Passive process, higher concentration moves. So when cells undergo metabolism or catabolism, or metabolism or anabolism or catabolism, they produce waste products. They produce CO2 and water. So as the CO2 builds up in the cell, it flows out of the cell by passive diffusion into the bloodstream, where it's absorbed by the red blood cells and brought to the lungs and gotten rid of. Okay? I don't know why this delays drive me crazy here. I don't know why it's delaying. Osmosis. All right, so that's diffusion. Then we got osmosis. Osmosis is a little different. It's a movement of water molecules from an area of high concentration of water to an area of low concentration of water. It's not the solute moving. Okay, it's not diffusion. Diffusion, the solute molecules moved. The solute are the molecules inside the water. In osmosis, the water molecules are moving. There's a semi-permeable membrane. The solute molecules are too big to pass it. So only water molecules are involved. So here you see water. Yeah, you look at all the blue dots are water. Okay. The green dots are your solute. Now, according to diffusion, the green dots should move in, but the green dots are too big to pass through the membrane. So what happens is water moves to equalize that concentration. Okay. So all the green dots, there's more green dots outside than inside, right? Everybody see that? Diffusion, the green dots would move in. These are too big to diffuse. So what happens is water is going to move. Water can pass through with a semi-permeable membrane. So what it's more concentrated outside than inside the cell. So water moves outside the cell to decrease that concentration, to make the concentration the same on both sides. That's osmosis. Okay? And it's very important in IV therapies and in red blood cells. Okay? In the cardiovascular system. Because red blood cells contain a semi-permeable membrane. If I can go back to where I was. All right, so this is osmosis. This is your semi-permeable membrane between the cells, right in here. And these are your solute particles. The solute particles cannot pass the membrane, so only water can move. So which way is water going to move? Is water going to move left to right or right to left? Water is going to move right to left. Okay? That's the direction water is going to flow. Okay? So water moves from a low concentration to an area of high concentration. The exact opposite of diffusion when you think about it. So this is osmosis. It's real important because our body functions osmosis because of the plasmalemma. So water moves from an area of low concentration to high concentration, so the concentrations are equal. As water moves to the left, what happens to the left side? The left side becomes more dilute, right? When the concentration is the same, diffusion stops. That's osmosis. Okay? And you get what's called tonicity in the cell. This is real important in red blood cells. So living cells contain aqueous solutions, and it's surrounded by aqueous solutions. Okay, the body's 66% water. Cell function requires maintenance of the same osmotic pressure inside and outside of the cell. If you give someone an IV and the osmotic pressure is different than a red blood cell, the red blood cell is either going to swell up and burst or it's going to shrivel up and die. Because water is going to move across that membrane. We couldn't figure out why transfusions were killing people. It was destroying the red blood cells.
You didn't give me.
Yeah, it's important to learn this before you give someone a transfusion. Okay?
Give me the pin.
So if the solute concentration of fluid surrounding the cells is higher, result what's called a hypertonic solution. That causes water to flow into your surrounding areas to make the concentrations match.
It's still the same one for that part too?
I'm sorry, what's that?
There was something different.
I'm sorry.
Oh, was there a question?
No, there wasn't. I apologize.
Oh, no problem. No problem at all. Okay, that's called crenation, where it's a hypertonic solution, water goes out, the cell shrinks up. Okay, solute concentrations are too low. It's a hypotonic solution. So hypertonic means there's more solutes outside the cell. Hypo means there's less. In hypertonic, water flows out of the cell shrinks up and dies. Hypotonic, water flows into the cell swells up and bursts. Either way, you're killing your red blood cells. And it is not good for your patient. If you wipe out the red blood cells, patients don't breathe. Isotonic solutions have ideal osmotic pressures. There's no osmotic pressure difference. The red blood cells stay happy and you can do a transfusion. Okay, so what happens if we place a red blood cell in a hypotonic solution? Okay, what's going to happen? Hypotonic means there's more concentration inside the cell. Which way is the water going to flow, into or out of the cell?
Out
It's going to flow into the cell
into the cell.
because the cell has more concentrated solute material inside. It's going to have to dilute it to the same concentration. Okay, there'll be a net movement of water into the cell. Remember, the red blood cells don't have the strength to exert sufficient pressure to stop the flow of water. The cell swells up and bursts. Okay, I'll do a drawing a little bit here. Alternatively, place the red blood cells into a hypertonic solution. Water will pass out of the cells and they will shrink. Everybody heard of Ringer solution? Ringer solution we use in hospital patients all the time. We hydrate them. Ringer solution is specifically designed to have the same osmolarity as a red blood cell. That's why we use it. I mean, it's composed of the balanced physiological, I was saying, in solution. Sodium chloride is 0.9 grams, potassium chloride is 0.042 grams, calcium chloride is 0.025 grams, and distilled water is 100 milliliters. That's how you make it. You're not gonna have to make it as a nurse, but you're gonna have to deliver this to patients. And that is exactly balanced to exert the same osmotic pressure as a red blood cell. Okay, there was a question here. So the first assignment that is due today, that's just an introductory how to use the system. You can do it or not do it. That doesn't make any difference. That's why there's 0 points assigned to it. Yeah, Ethan had it exactly right. It's just a tutorial basically to help you understand how the program works. Okay, that's why it's assigned like that. That's not giving you points on that one. All right, so that's your Ringer. So that's why we use Ringer solution. Now, let me. Okay, so there's your red blood cells. Let me get out of this and go to, and maybe draw a little picture. Sometimes that'll help understanding if I can draw it a little bit better. My drawings are not that great, as you can see my memory. All right, so this is a red blood cell. It has a membrane that's going to allow certain things to flow out and certain things not to flow out. These are your particles. If we put a solution which has less particles in here, which way is water going to flow to make the concentration the same? Remember, it flows from lower concentration to higher concentration. So water is going to flow into the cell. And the cell swells up and bursts. This is a hypotonic solution. Okay, does everybody understand? It has to make the concentrations the same. And you can't move the soluble particles. They can't fit. They can't go through the membrane. So the only thing that can move is water. So water flows in to make the inside concentration lower till it matches the outside concentration. As water flows in, the red cells will expand and burst. A hypertonic solution. Okay, here's your red blood cell. And these are your particles. Now outside the cell, we have all these particles. This is hypertonic solution around it. Which way is water going to flow? It's going to flow to make the outside as dilute as the inside. So water is going to flow out. Perfect. Water flows out. Okay. That causes crenation. The red blood cell shrinks up and becomes non-functional. In an isotonic solution, this is your red blood cell. And this is your solution. Okay. They're the same. So water flows in and water flows out at the same rate. Okay, nothing changes. There's not a net change overall. So in a hypotonic solution, and you'll see this on your nursing exams when they're doing Ringer solution. If you're given a hypotonic solution, the red blood cells will swell up and expand and explode. Okay, water will flow into the red blood cell. In a hypertonic solution, water flows out of the red blood cells to get the concentration the same, and your cell shrinks up and dies, called crenation. In an isotonic solution, which is Ringer solution, it's specifically made for that, so you can give it IV and not kill your patient, which is a good thing, right? Unless you don't like your patients. Okay, and an isotonic, I didn't put isotonic. This is an isotonic solution. Okay, there's no net change and the patient survives. So that's the difference between hypotonic, hypertonic, and isotonic solutions. Hypertonic, water flows out to make it equal. Hypotonic, water flows in. Water flows out, you get crenation, cell shrinks up. Water flows in, you get hemolysis. Lysis means explodes. Heme means blood. So the red blood cell explodes. Okay? Isotonic, everybody's happy. No net flow of water. Flows in and out at the same rate. Okay, any questions with that? Does that make sense?
Yes, thank you.
All right, perfect. All right, this is a normal red blood cell. That's a healthy red blood cell. That's what it's supposed to look like. It's biconcave, a lot of area for absorption of oxygen. And the computer's gonna make it really slow doing this. This is crenation. This is when the red blood cells condense, water flows out. This is a hypertonic solution. That's what a red blood cell looks like, crenation. Again, looks a lot different, right? That's not gonna function. That's not gonna transmit oxygen. Okay, and that's when the red blood cell swells up. Whoops, go back. This is when the red blood cell swells up. You see how it's lost its biconcave shape. It's all swollen, and they start to explode. And these are little bits that are left after the cell explodes. Okay, so osmotic pressure is really important with red blood cells. Now, osmotic pressure is determined, okay, like with in chemistry. Again, water is going to flow to an area of low concentration to high concentration. Exact opposite of diffusion, because the solute particles can't move. Okay, and it's going to cause that side to rise. Water flows in, that column's going to go up, right? As that rises, the force required to push that back down to normal, that's osmotic pressure. That's it. Water flows in, it makes that column go up because it's putting more water on that side of the glass. The force it takes to push that back down so the water levels are equal is like osmotic pressure. That's the definition of osmotic pressure right there. That's what you need to remember for it. All right, so membrane permeability. You also have, so you have three types of passive diffusion. You have diffusion, you have osmotic pressure, and you have facilitated diffusion. And passive means what, remember? Passive means no energy required. Okay, so passive processes facilitate diffusion. Solutes are passively transferred by a carrier protein. In other words, it bonds to something, and that allows it to move through the membrane, a concentration gradient, and be released on the other side. So a concentration gradient are size, charge, temperature, number of carrier proteins affect the rate. Glucose and amino acids are involved in transporting things across the plasma lemma. So things that normally can't go through the fatty acid part of the membrane bind to something that allow it to transport across. Okay, like glucose has channels that allow glucose to go from outside the cell to inside the cell, right? We have to get glucose into our cells. That's what our main energy source is. So if something can bind to the glucose and piggyback to it, it's going to go through that with the glucose and not using extra energy. That's what facilitated diffusion is. It's bonding on to a carrier protein, and it's kind of like just binding on, it's getting a free ride. And then when it gets inside the cell, it releases. That's facilitated diffusion. All right. Every once in a while this binds up on me. Okay, so we talk about facilitated diffusion, and you can see glucose coming in and glucose going through. So facilitated diffusion, solute molecules transport across the plasma membrane as a carrier protein. So it binds to the glucose, and this little molecule comes right across. As it goes through this channel, as the glucose goes through it, it goes through with it. And it allows it to come right across and be released into this internal part, into the intracellular fluid. Okay, active transport. Active transport is the opposite of passive transport. Active means it requires energy to do. Okay, there's three types. Active process for is active transport. Solute molecular transport concentration regardless of the concentration gradient. Okay, sodium potassium pump with our nerve signals, it has to go against the concentration gradient to keep that established. So when the nerve fires, it can switch sodium to potassium. So that's an active transport to keep that up. So ATP is used. ATP goes to ADP, gives off a phosphate group that releases energy. That's how our body works. Energy is stored, okay, as ATP for usage. When we use ATP, that's energy being produced. So active transport requires ATP. Sodium, potassium, calcium, magnesium ions are all involved in that kind of transport where it's actively transported. Okay, so here's your transport system. Uses ATP, goes to ADP. If you can see my little mouse cursor there. Okay, and you take potassium going from outside to inside. You take sodium going from inside to outside. It pumps it against a concentration gradient, so it cannot be passive. You're going against the concentration gradient, so it's an active transport system. It requires ATP. That way, when a nerve fires, sodium goes in, potassium goes out, and the nerve propagates, and you get your signal. When it rests, sodium goes back out, potassium goes back in against the gradients and reestablish what's called your action potential. We'll go through a lot of detail on that. All right, does that make sense? That's active transport. There's two other types of an active transport. Okay, there's pinocytosis and endocytosis. Okay, it's an active process. Endocytosis, there's two types of endocytosis. There's pinocytosis and phagocytosis. They're the same thing except pinocytosis is small, phagocytosis is larger. Same thing. Vesicles bring small molecules into the cell. A variety of stimuli affect the movement. Extracellular fluid is involved. It uses ATP. Phagocytosis brings solid particles, larger ones. So endocytosis brings things into the cell. Endo means internal, like an endoskeleton, your internal core. So endocytosis means it brings things into the cell requiring energy. If it's pinocytosis, it's small. If it's phagocytosis, it's larger ones. Okay, that's the only difference there. On cells, slide change. Okay, you can see here, pinocytosis, you got something in the cell, it forms around it, it takes it, fuses with the plasmalemma, and kicks it out of the membrane. Okay. Phagocytosis, it fuses with a membrane around it, comes to the membrane, fuses, and kicks it out. Okay. Pinocytosis is a vesicle that secretes and releases a small particle. Phagocytosis incorporates it, fuses with the membrane, opens up, and secretes a larger particle. So phagocytic vesicle is a larger particle. Pinocytosis is a small vesicle. Okay? But that's how it gets out of the cell. So you got active transport and you got endocytosis. The other active process is exocytosis. Now, without looking at the slide, if endocytosis means bringing it in, exocytosis means what?
Bringing it out.
Pushing it out. Perfect. Perfect. He's pushing it out. Okay. It's the release of intracellular material to the extracellular environment. Requires ATP because it's an active process and requires calcium ions. Okay. Fluid and cellular waste, our secretory products are involved in doing that. Okay. So your three processes requiring energy of active transport, endocytosis, which is two types, and then exocytosis. All right. And here's exocytosis. You have material, you form a membrane around it, you push towards the outside, it fuses with the plasmalemma. Instead of bringing it in, this time you're pushing it out. Okay, so endocytosis brings it in, exocytosis brings it out. Both processes use ATP for it to be accomplished. That's why they're active processes. All right, extensions of the plasmalemma. Remember we talked about microvilli. Microvilli have several functions. Two of the most important function is increase in surface area. That allows for increased absorption and increased secretions. They're finger-like projections of the plasmalemma. They absorb materials from the extracellular fluid. They increase the surface area of the plasmalemma. They can bend back and forth in a waving manner. This movement helps to circulate extracellular fluid and bring nutrients to it, brings waste away from it. Okay? Don't get microvilli confused with cilia. Remember we talked about cilia in your lungs. Those only beat in one direction. Microvilli beat in both directions. Okay, the cilia are a lot longer than the microvilli and a different function. They beat mucus and contaminants, things in the lungs, out of the lungs. Here's it. That delay again. All right. Cytoplasm. Remember, the cytoplasm is inside the cell, the plasmalemma. There's two parts. There's a cytosol and these organelles, both membranous and non-membranous. The cytosol is your intracellular fluid. That's the liquid part. It consists of your nutrients, your proteins, and waste products. Okay? So your cytosol is where your oxygen gets dissolved into. It's where your carbon dioxide gets dissolved into when it goes out and oxygen comes in. Organelles, those are intracellular cells that perform specific functions. We talked about mitochondria, Golgi apparatus, lysosomes, peroxisomes. We showed you what those were. And then some are membranous, some are non-membranous. Okay. Cytosol, this is the liquid portion of the intracellular fluid. Contains a higher concentration of potassium ions and a lower concentration of sodium ions as compared to the extracellular fluid. That allows our nerves to function. The sodium-potassium pump, remember, it's an active process, active transport. It pumps sodium out of the cell, potassium into the cell, and creates a potential differential, allows our nerves to fire. Consists of a net negative charge, contains a high concentration of protein, contains a small quantity of carbohydrates, contains a large reserve of amino acids and lipids, which we use to make our proteins, make our steroids and things like that we use, and contains large amounts of inclusions, different things within the cell. Okay, that's your cytosol. That's the liquid part. Okay. And the cytoplasm. Cytoplasm has your organelles. It consists of your cytosol, which was your liquid part, and your organelles. The non-membranous organelles are your cytoskeleton, your microtubules, your microfilaments, your centrioles, which are used in cell division, the cilia, the flagella, and the ribosomes. Membranous organelles are your mitochondria, your nucleus, your endoplasmic reticulum, your Golgi apparatus, your lysosomes, and your peroxisomes. And remember, when you have your endoplasmic reticulum, there's two types. There's rough and there's smooth. Rough means what are attached? Ribosomes, right?
Ribosomes.
Perfect. Ribosomes. If ribosomes are attached, it's rough endoplasmic reticulum. If ribosomes are not attached, it's smooth endoplasmic reticulum. Rough endoplasmic reticulum makes your proteins. Smooth endoplasmic reticulum makes your cholesterol and your fatty acids. Okay. The cytoskeleton consists of your microfilaments, your intermediate filaments, your thick filaments, and your microtubules. Those are all structures like a scaffolding that hold the structure of the cell and determine its shape. Those are all the different tubules of it. Okay. Thick filaments. Thick filaments are composed of myosin. Okay, the myosin protein. Myosin is used in muscle contractions. Okay, we'll get into that a little bit later on down the line too. But your thick filaments are your myosin. It's found in muscle cells and is involved in muscle contraction. Okay, microtubules, composed of tubulin proteins. They're involved in the formation of your centrioles that we use during cell reproduction. They move duplicated chromosomes to opposite poles of the cell, so the DNA gets divided evenly with each daughter cell. Right? They're involved in anchoring organelles. They have the Golgi apparatus and the peroxisomes and lysosomes all have to stay in the certain spots. They anchor them into those positions. Okay, they perform a function during cell reproduction. Okay, they're involved in moving cell organelles, involved in moving the entire cell as the cell can for motion, and they'll move material across the surface of the cell. Those are what your microtubules do. Okay, you can see the different skeletons here. You can see the thick layers here. You can see all the fibers. Those fibers are your microtubules, your microfilaments, and your tubulin proteins that determine the shape of the cell and actually allow the cell to take different shapes and actually allow it to move. Okay, here's your typical cell. If you look at the cell, you have your microvilli, which are your extensions. Underneath you have your microfilaments to support those extensions. The plasmalemma is your membrane. You have a web of those fibers. You can see a mitochondria in there. You can see intermediate filament fibers. So your endoplasmic reticulum. Is that endoplasmic reticulum rough or smooth?
Rough.
Rough. Perfect. It's rough because it has ribosomes attached to it. Okay. You can see microtubules and those secretory vesicles. Remember those little secretory vesicles like with endocytosis, where it takes it and moves along the membrane, it follows these little microtubules along the track till it gets to the membrane. Okay. Non-membranous organelles are your centrioles, your cilia, and your flagella. These are different, there are different descriptions of them. You see your centrioles, and it's microtubules. They're in the centrioles, they're near the nucleus, and they're spindles that move the chromosomes during cell division. The cilium are little microtubules also the cell surface, and these propel fluids and surfaces across the cell to move waste away, move nutrients to it. Flagellum are found on sperm cells, and they propel the sperm through the vaginal fluid to reach the egg. Okay. Ribosomes. Okay, these are non-membranous organelles. Free ribosomes float in the cytoplasm. Fixed ribosomes attach to endoplasmic reticulum. Both produce proteins. That's what ribosomes do. Okay. Membranous organelles. There's a double membrane organelles. Your mitochondria, which produces ATP. 95% of all ATP is produced in mitochondria. The nucleus, which contains your chromosomes, your DNA. Endoplasmic reticulum, which is a network of hollow tubes. If it has ribosomes attached, if it's rough, then it produces proteins. Those go through these tubes and get modified in the peptide. If it's through the endoplasmic reticulum, it produces your cholesterol. Okay? Golgi apparatus. After the endoplasmic reticulum, it moves to the Golgi apparatus, which modifies the proteins. Your lysosome contains cellular digestive enzymes to break things down. Your peroxisomes contain cellular digestive enzymes to break things down, and it's specifically designed to help break down hydrogen peroxide. The name peroxisome, therefore. Hydrogen peroxide is extremely toxic to cells. Peroxisome is designed to destroy hydrogen peroxide. Mitochondria, that's what makes your ATP, has what's called cristae and consists of a matrix. Okay? The cristae are the divisions within the mitochondria that form different chambers, basically. Okay? You have an inner membrane and an outer membrane. Okay, it uses oxygen and produces CO2. And in the process, ATP is produced. So glucose and oxygen produce CO2 and water, and ATP is produced in the process. That's what a mitochondria looks like. And again, this forms these little channels inside of which are called the cristae. Now, the nucleus is the control center of the cell. It gives you all the directions. Okay, there's a nuclear envelope around it. Nucleoplasm is the inner part, just like cytoplasm is inside the cell. Nucleoplasm is inside the nucleus. You have a perinuclear space, is the space between the inner and outer membrane of the nuclear membrane. Remember, it's a double membrane. You have nuclear pores. Nuclear pores allow the DNA to make RNA and send it out into the cytoplasm where the ribosomes are to make proteins. And you have a nuclear matrix inside the DNA. Okay. And here's a picture of it. You see you have your nuclear pores. You see the nuclear envelope. The nucleolus is the center. Again, DNA is your chromatin. Most of the time, DNA is all spread out. It's not in chromosomes. It only forms chromosome and binds that when it has to duplicate and split into different cells. But it's usually this chromatin, which is all spread out, so it can transmit into RNA and it can be read. You have the perinuclear space and the nucleoplasm. And again, on the bottom left here, you can see these nuclear pores, which allows RNA to transfer information from the nucleus into the cytoplasm to produce the appropriate proteins that the cell needs. And that's a scan electron micro picture of it. You see some of the pores and the double layered nuclear membrane. Okay, chromosomes, when the cell gets ready to divide, the chromatin starts to shrink up and wrap around what's called histones and form these little condensed areas of chromosomes. Chromosomes are DNA wrapped around protein called histones. The nucleosomes in it and a chromatin. So chromatin, nucleosomes, and then chromosomes. As it condenses more and more and more, it forms into these chromosomes. And this is what it looks like. This is your chromatin. This is how DNA usually is, so it can be read unzipped, the double layer, and one side read. As it starts to coil up, you see the DNA process come here, it starts to coil. And you see as it winds up, it rounds around these proteins here. This is a nucleosome. These are histones, these little protein balls that it used to wind up around it. And it forms a nucleosome. As it gets tighter and tighter, it forms a chromosome. And these chromosomes are connected in the center region by kinetochore. Okay, is the protein here. And the centromere is the center part of the chromosomes. And when the cell divides, one chromosome, the centromere splits, and one chromosome goes to each cell, each daughter cell. So chromosomes are the visible part of DNA. Chromatin It's basically the all uncoiled part of DNA. Whoa, went too far again. Went too far that way. Okay, endoplasmic reticulum, ER. Everybody should know this by now. Two types, rough endoplasmic reticulum, smooth endoplasmic reticulum, just by the presence of ribosomes. Rough endoplasmic reticulum consists of fixed ribosomes, and proteins enter into the endoplasmic reticulum, into those channels, and it modifies the protein as it goes through it. Okay, this is what rough endoplasmic reticulum looks like. This is what smooth endoplasmic reticulum looks like. This has all the ribosomes. This doesn't have ribosomes. Okay, you can see the nucleus is close. Okay, goes through the nuclear pores, and here's your endoplasmic reticulum right by the nucleus. That way the RNA doesn't have to travel far to be translated into a protein. Okay, ribosomes consist of two units, a large subunit and a small subunit. They exist apart. When RNA comes in there, it comes between, these two come together and squeeze the RNA into a reader, and it reads it as it pushes it through like a Xerox machine. Smooth endoplasmic reticulum. Remember, rough endoplasmic reticulum synthesizes proteins. Smooth endoplasmic reticulum synthesizes lipids, steroids, carbohydrates. There are also stores of calcium ions, and there's also some detoxification of toxins inside the smooth endoplasmic reticulum. Okay, but the big thing to remember is rough endoplasmic reticulum, proteins. Smooth endoplasmic reticulum, your lipids, your steroids like cholesterol, and your carbohydrates. So membranous organelles, the Golgi apparatus. Remember, the Golgi apparatus is after the proteins are synthesized in the endoplasmic reticulum. They travel to the endoplasmic reticulum. Their next stop is the Golgi apparatus. So they're transmitted by little vesicles, little vehicles, from the endoplasmic reticulum into the Golgi apparatus. So some modification occurs in the endoplasmic reticulum, and a lot occurs in the Golgi apparatus. So the Golgi apparatus synthesizes and packages of your secretions. So it gets the enzyme, the hormone, whatever it's needed, packaged and sent and activated so it can be released into the blood, into the extracellular fluid, or used by the cell. It packages so it goes to the right place. That's what the Golgi apparatus does. Packaging of enzymes, so it can be released into the bloodstream, so it can go to the target organ. Renewal and modification of the plasma lemma. It rebuilds your plasma lemma. Things break down. You got to replace it. It comes from the Golgi apparatus. All right. It's got a mind of its own sometimes. Oh, there we go. And back up. Okay, this is what the Golgi apparatus looks like. It looks like a series of pancakes. The flat end, the really wide end down here, this is where the protein enters from the endoplasmic reticulum. You can see little vesicles. You can see my little pointer here. Okay, there's my pointer, and these are the little vesicles right here. These are coming from the endoplasmic reticulum. So they enter what's called the cis side. The trans side is the smaller side where it's released. So it enters the cis side, the big flat side of the pancake. It goes through all these different intervals, just goes winding around through it, and it gets modified. It gets changed. Okay, then it goes to the end, which is the trans side, and then the end of the Golgi apparatus surrounds the protein and breaks off. And now you got little secretory vesicles. This little secretory vesicle either goes to the plasma lemma by following the fibers, actinomyosin fibers of the cytoskeleton, and goes to the membrane and gets released, or it stays in the cell and gets used. Okay, but that's what the Golgi apparatus does. Come on, shift. It's going to shift a whole bunch at once again. There we go. All right, so that's the relationship between the RNA comes out of the nucleus into the cytosols, into the endoplasmic reticulum, into the Golgi apparatus, out of the cell, into the secretory vesicles. Lysosomes, we talk about lysosomes and peroxisomes. They eat things. They dissolve bad things, okay? That's what they're designed to do. Now, they can be used for autolysis, which is a self-destruct button for the cell. If the lysosome peroxisome ruptures inside the cell, that's autodestruct. It means the whole cell dies because it's full of digestive enzymes. So you want to make sure, you got to remember a lysosome peroxisome is a membranous organelle. It has to be. If a membrane wasn't around it, all the digestive enzymes would come out into the cell and they'd autolyse. The whole cell would self-destruct. So lysosomes, they actually fuse with the phagosomes and digest solid material. So pathogens, things like that. Bacteria, they'll fuse with it and digest it. They recycle damaged organelles. If your aminos start to fail, the lysosomes come, dissolve it, and we can reuse those parts again. They sometimes rupture, killing the entire autolysis. If they rupture, they'll kill the entire cell because all the designations get turned loose. Okay, peroxisomes. Peroxisomes are just like lysosomes, except they're specifically designed to convert hydrogen peroxide to water and oxidants. Hydrogen peroxide is extremely toxic to the cell. So you have a special structure designed just to handle that. But they're the same thing as peroxisomes. They look the same. It's their designation is to help get rid of hydrogen peroxide. They consist of catalysts. They're abundant in liver cells where they're found. And they convert hydrogen peroxide to water and oxidants. Okay, membrane flow, continuous movement of recycled cell membranes. Your membrane has to be replaced, your plasmalemma. Transport vesicles connect the endoplasmic reticulum with the Golgi apparatus, like we said. Secretory vesicles connect the Golgi apparatus with the plasmalemma. So if the plasmalemma needs protein to fix its cell, okay, it goes from the endoplasmic reticulum, vesicles to the Golgi apparatus, up to the plasmalemma. Vesicles remove and recycle segments of your plasmalemma to keep it all intact. Here's a perfect picture of it. Okay, at number 1, there's your messenger RNA. Your messenger RNA feeds into a fixed ribosome, which is attached to the plasmid reticulum. As a ribosome makes the protein, it takes amino acids and attaches them into amino acid chain. It links them together. That's what proteins are. They're a series of 20 amino acids, and it links them together. And as it links together, you can see how it travels through the endoplasmic reticulum, and it gets modified. When it reaches the end of the endoplasmic reticulum, it goes into a transport vesicle. It breaks off and feeds into the cis form, the cis face of the Golgi apparatus, where your vesicle fuses with the Golgi apparatus, and now your protein's in the Golgi apparatus. Okay? As it goes to the Golgi apparatus, it goes through the Golgi apparatus and works its way through these different layers. It gets more and more and more until it gets into the other side. Once you get to the other side, you get another secretory vesicle. It breaks off and either can go to a lysosome or go to something in the cell, or that vesicle can go to the plasmalemma and help replace that, or it can go to the plasmalemma and fuse with it and release it into the bloodstream to go to other cells or other organs where it's needed. This is a perfect diagram of how everything works. It's just picture form of it. Okay, let's do this and we'll call it a night. Let's see if you know how much you gotta remember. Okay, here's where we're starting. What's that pointing to? No cheating.
Golgi apparatus.
Golgi apparatus. Perfect. We're going down. What's the next one?
Mitochondria.
Mitochondria. What's the next one?
Ribosome.
Lysosome or peroxisome. Either one, they look the same. But that just, I mean, it's purple in the picture, but they look the same on a cell. The little bug right there. There he goes. Okay, what's the next one pointing to? It's looking at the little holes in the nuclear membrane.
Nuclear pores.
Your pores.
Nuclear pores. Perfect. And the next one.
Smooth ER.
Perfect. No one should miss that. Smooth ER. And the next one is
Rough ER.
rough ER. You got it. And this is pointing to the little dots.
The attached.
Yep, attached or fixed ribosomes. Perfect. And little dots that are by themselves.
Ribosomes.
Chromosomes.
Free floating or unattached, right? Free ribosomes. Okay. And next is I'm going to the left now and down. What's that? That flap.
Plasma membrane?
Plasmalemma. Yep, your membrane, cell membrane, plasmalemma. Perfect. And the next is points to little fibers.
Cytoskeleton.
Cytoskeleton for microfibers. And the next one is your,
The space.
it could be the perinuclear space. Is that what it is? Or nuclear envelope, same thing. It's hard to tell what it's pointing to. The next one is pointing to right dead center of the nucleus.
Nucleus.
The nucleolus. That's the center of the nucleus.
Yeah.
It's the nucleolus. Okay. What's the stuff inside the nucleus, the liquid stuff?
Is it nucleoplasm?
Nucleoplasm.
Yeah,
What's the stuff that's kind of
nucleoplasm.
unraveled? What's the DNA that's unraveled?
Chromatin.
Chromatin. Okay. The next ones are your
Centrally?
centrioles. Okay. And the next one is your,
Lysosome.
yep. Okay.
Centrosome.
Centrosome. Central and centrosome look very familiar, and they're only used in cell division. And the next one is, if the other one was a peroxisome, this one's a what?
Cytosome.
Lysosome. Right?
Lysosome.
Because it's the same thing as a peroxisome, just because dissolves hydroperoxide. And the liquid portion is called your
Cytosol.
cytosol. And what happens when it breaks off the Golgi apparatus? Those are what?
Proteins.
Not proteins. Proteins are inside of them. What's the container called?
Vesicles.
Vesicles. Perfect. The proteins are inside of them. Those are secretory vesicles with the proteins inside. And the little hairs on top.
Microvilli.
Microvilli. Perfect. Remember anchoring junctions? We also have what's called focal adhesions. Those connect intercellular microfilaments to the protein fibers. They're found in epithelial tissue that migrate and were used during wound repair. So the filaments that line up so your skin can come back together during a cut or some kind of trauma. Hemidesmosomes are connecting cells that are exposed to a lot of abrasion, a lot of force. Examples of the cornea of your eye, skin, vaginal tissue, oral cavity, esophagus. And you're going to have a constant abrasion. You're going to have hemidesmosomes to help protect those areas that hold those cells together. Okay. This is an example of a hemidesmosome. The hemidesmosome has two layers in your basal lamina. The basal lamina is where it's held together to the body. Has a clear layer towards the surface of the cell, and then a dense layer towards the next cell. So hemidesmosomes attach an epithelial cell to extracellular structures like protein fibers, like in the basal lamina, to help hold the cell together into its position. That's what separates the hemidesmosomes from the rest. All right, you got the cell life cycle, how a cell reproduces. Okay, cell reproduction consists of special events. You need to know the events and the order they occur in. You have interphases when the cell does all its housekeeping. All it's making is proteins, hormones, whatever it's making, it just keeps making those. Then it has to divide. It goes through 4 phases of cell division. There's prophase, metaphase, anaphase, and telophase. If you want a mnemonic, it's PNET. Prophase, metaphase, anaphase, telophase. It gives you the order of those that occur. Then cytokinesis is anaphase and telophase, where it actually breaks apart and the cell forms 2 daughter cells. Sometimes you want for the slides to switch. It does every once in a while. Oops. Okay. So interphase, we said everything in the cell is duplicating. It's G1 as in G2 phases. You get duplication of the organelles and protein synthesis, chromosome replication and DNA synthesis and histone synthesis. Remember, the histones are the stones that the DNA wraps around as it goes from chromatin to chromosomes, okay, as they become more compact. Okay? And you got protein synthesis going on. So it's doing all the housekeeping, what the function of the cell is supposed to do to get ready to multiply and divide. Okay. DNA is read in segments. When it's in chromatin, the DNA is separated. So it can unzip and they compare different bases. And you can see here how DNA duplicates, getting ready for cellular division. It makes a copy of itself. So the DNA unzips and all these bases have complementary bases that they use to make a duplicate. So adenine, you have four bases for DNA: adenine, guanine, cytosine, and thymine. Okay, adenine always pairs with thymine. Guanine always pairs with cytosine. So when this opens up right here, if you have adenine here, you know you got a thymine here. If you get guanine here, you know you got cytosine here. Okay? Again, guanine, cytosine. And these all link up and these chains get filled in, and it makes an exact duplicate of the DNA strand. So that way when the cell splits, you get an exact replica of the DNA. That makes sense? Okay. That's how it gets duplicated. So cell reproduction is called mitosis. Okay. Mitosis consists of PMAT, prophase, metaphase, anaphase, telophase. Prophase is the first phase of mitosis. Begins after DNA replication is complete. So after the DNA has unsplit, replicate itself, come back together, then prophase can begin. And it starts to form chromosomes. Metaphase is when the paired chromatids line up in the middle of the nuclear region. Okay, so prophase is DNA replication is complete and you start to form chromosomes. Metaphase is when they line up in the middle so they can be split apart. Anaphase is when the paired chromatids, the chromosomes, separate to opposite poles of the cell. They're pulled apart. And telophase is the two new nuclear membranes begin to form. You start to get cleavage between the existing cell and it actually starts to split the cell apart. So here is, if this pulls up. So here's interphase. You watch the mouse pointer. That's where the proteins are being synthesized. This is prophase. Prophase is where all the DNA is duplicated. And you see how much darker it becomes in the DNA becomes the nucleus here. It's starting to coil and form chromosomes. Okay, that's prophase. And then the chromosomes line up in the middle of the cell. Okay, metaphase, they line up dead center in the middle, and you can see the spindles that are starting to attach so they can be pulled apart. Anaphase is when you actually see the chromosomes being pulled apart. And telophase is when you have cleavage between the two cells and two new daughter cells are starting to form. Cytokinesis, when the cells completely separate, and now you have two new daughter cells. So prophase, metaphase, anaphase, telophase. Prophase is DNA forming chromosomes. Metaphase is the chromosomes lining up in the middle. Anaphase is the chromosomes pulled apart, or one goes to each daughter cell. And then telophase, when the cell starts to invaginate the plasmalemma, called cleavage, and break into two cells. That's your series of steps right there for mitosis. So cytokinesis, the division cell, the cell member begins to invaginate to form two new cells. Many times this phase actually begins during anaphase. This is the conclusion of cell reproduction. Once the cells separate, cell reproduction is complete. Mitosis is complete. Okay, this is what it looks like as far as time-wise goes. So from prophase, metaphase, anaphase to telophase, you're looking between 1 to 3 hours in time frame. The rest of the cell cycle is interphase, when the cell does all the basic housekeeping stuff and what it's supposed to be doing.